Good morning and welcome to the intersection of art and science, using 3D animation to tell the stories of biology with award-winning biomedical animator, Dr. Maya Diviak. My name is Anne Smith and I'm the Artistic Director of the Arts Learning Festival at Independent Schools Victoria, and I have the pleasure of introducing this morning's program to you. The Arts Learning Festival is a biennial event open to all schools and community to celebrate the power of the arts to teach, to connect, and to inspire us all. Before we begin, I'd like to acknowledge the traditional owners of the land on which we meet today, wherever we may be, and I pay my respects to their elders and all First Nations people, past, present, and future. And now to this morning's program. I'd like to begin by recognising the partnerships that have brought us together this morning. Our wonderful hosts here at ACME, my colleague Gary Westmore, who has provided the space and technical requirements to ensure that this event is delivered seamlessly to you wherever you are. And to the Peter MacCallum Cancer Centre, where our guest speaker, Dr. Maya Diviak, is the inaugural biomedical animator, where the works she creates are about the cellular and molecular mechanisms leading to cancer. Maya completed her PhD in molecular biology at Monash University before moving into scientific sales. A chance meeting with world-renowned animator Dr. Drew Berry at the Walter and Eliza Hall Institute opened the doors to the wonderful world of bioanimation. Maya then pursued her dream of becoming a biomedical animator, studying multimedia at Swinburne and 3D animation at Australian Film, Television and Radio School. She then won a coveted animation fellowship with Dr. Drew Berry and has been animating professionally since 2013. Her creations have been exhibited at the Vivid Festival of Light, Music and Ideas, and she has won several awards. These include a Platinum Remy Award at the WorldFest Houston International Film Festival, the Medical Education Award in Motion Media at Bioimages, the Jury Award at the Raw Science Film Festival, Awards of Outstanding Excellence at the Doctors Without Borders Film Festival, and official selection at the Melbourne Documentary Film Festival. And so before Maya begins, if you do have questions, please type them into the chat function during the presentation and we'll try and answer as many as possible at the end. Thanks very much, Maya.
Thank you, Anne, for that wonderful introduction, and thank you to ACME and Independent Schools of Victoria for the wonderful opportunity to speak to you today. Don't seem to be working. Oh, here we are. Okay, we're back in action. Yes, I'd just like to begin by acknowledging the traditional owners, the Wurundjeri people of Kulin Nation, on whose land we meet, share and live today. So let's dive right in. Let's begin with some perceptions of science. Number one, science is boring. And over on the right there, we have that very conservative Victorian-looking gentleman saying, a lot of people think science is boring, but is it boring that nucleic acid is a macromolecule composed of chains of monomeric nucleotides? Pretty fascinating, huh? Well, if you're not a scientist and you don't understand that language, yes, that will sound pretty boring to you. Science is complicated, and here we have Dr. Kong Hui Yao in front of her whiteboard with a very complex looking series of diagrams, arrows, molecules connected everywhere, graphs, and so on, all explaining cancer metabolism. And that looks incredibly complicated. Number three, scientists are nerds. And in the bottom right-hand corner, we have self-confessed bio-nerd Risha Chakraborty, who is a secondary school science enthusiast. So why are these perceptions a problem? These ideas, and you may have thought these yourself or even said them, these ideas shut us off from the interesting world around us. So the environment around us and the interesting world within us. We have a whole universe of molecules inside our body, a whole universe of cells inside our body, and then a whole universe of molecules inside those cells, all moving around, jostling about, communicating and interacting, without us even having to think about it or even being aware of it. And these molecules allow ourselves to function, to communicate, to divide, to repair, and eventually to die. And so these molecules are the basis of life. And this is what I'm fascinated with, the universe of molecules inside ourselves. So perhaps the solution lies in better communication about science or better representation of what scientists are actually like. So can biomedical animation change the way people feel about biological sciences? And this is the story that I want to tell about with biomedical animation. And this is what's important to me. So let's start by defining what biomedical animation actually is. So biomedical animation uses 3D animation techniques from cinema, so throughout the movies that you've been watching your whole life, to explain biology. And in so doing, it accelerates understanding because it unfolds before your very eyes. It interests people in science because they can understand it, because they can see it, and it shows us just how beautiful biology is. And you can see from those 4 images there that biology, and in particular molecular biology, is incredibly beautiful. And I feel so lucky to be able to get out of bed each day and tell the stories of these molecules and bring them to the world. So why is biomedical science, biomedical animation important? You're probably aware that 60% of our brain is devoted to processing visual information or performing vision-related tasks. So we're visual creatures, and we have been since the days we first scanned our environment in search of food. But these days, with the development of the internet and the rise and domination of social media, we're becoming even more visually focused. So visual approaches are therefore the best way to interest audiences in science. The other important thing it does is connect the research laboratory to the public. So many people think that scientists are these really weird people who wear white lab coats and they do strange, inexplicable things behind closed doors with their tax dollar. And we really need to open the lines of communication to better explain what it is that scientists are actually doing. And biomedical animation does this. It provides a window on the research being conducted in the lab. And thirdly, it's important at the Peter Mac because it enables patients and people without scientific background to understand molecular processes. So as explained, we have a whole universe of molecules inside our cells, and biomedical animation makes us aware of this molecular world and connects us to the drama of our biology that can sometimes go completely off script, leading to situations such as cancer. It's also incredibly useful for explaining complex cancer treatments. So for me, biomedical animation is all about inclusion. It's about bringing those without any scientific background into the world of biology. And even if they don't engage with the science or necessarily understand it, that they can appreciate the beauty of it. But why is it really important? DNA damage is well recognised as a critical factor in the development and progression of cancer. So you could describe DNA damage like this. DNA damage consists of single-strand breaks, which are discontinuities in one strand of the DNA helix, double-strand breaks, where both strands of the DNA duplex are severed, DNA kinks, which occur when stacked base pairs become unstacked, causing DNA to make a sharp turn, and pyrimidine dimers, which cause 2 separate DNA helices to stick together. And this is the sort of response you're going to get. The person is going to look at you with a completely confused expression. They're going to turn around and walk away and wonder, well, why did I even bother? Or you could show them this. . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . .
DNA encodes the information needed to make proteins, which are the building blocks of our body. To keep 2 meters of DNA organised within the nucleus, it is wound around special structures called histones, much like thread on a spool. Damage to DNA can include single-strand breaks, double-strand breaks, changes to the DNA code, DNA kinks, and DNA sticking together. It has been estimated that an individual cell can suffer up to 1 million DNA changes per day.
And you can see that that's a completely different situation, can't you? Visual representation of this really complex biology makes it so much easier to understand. And it's also intriguing. People enjoy looking at it and they remain engaged. So let's take a deep dive into what creating a biomedical animation actually involves. You need to begin by deciding on your animation project. And at Peter Mac, I'm really lucky because we're not only a world-leading specialist cancer hospital, we also have an integrated world-leading cancer research program within that hospital. So I'm able to select from the most interesting and cutting-edge projects that we have within the Institute and animate them and bring them to the world. So I consider myself very lucky in that regard. You need to talk to the scientists if they're available, and of course at Peter Mac they are, they're all there doing their research. And you need to do this in an ongoing way throughout the project to ensure that everything is on track and that you're representing their story correctly. You need to read a lot in order to get your head around the subject, to be able to understand it and then distill that into a visual format. And in order to ensure that I do understand it, I like to write a literature review and outline everything in sequence, the story. I will then typically distill that literature review into a visual readout in the form of a storyboard. And you can see an example of that over in the bottom right there. And this looks a lot like a comic strip with the shots in sequential order. You will then show this to the scientists, of course, and ensure that they're happy with the story as we've laid it out. I then like to write a script, and I find the script really handy for controlling the flow of the animation because you're able to time the length of each shot according to what you need to say to explain each shot. And that just makes everything flow really nicely. You then need to assemble your models from a freely available repository called the Protein Data Bank. This contains a couple of 100,000 protein and DNA structures. However, you won't find whole molecular assemblies. You'll find little bits and pieces and you'll have to put them all together like a jigsaw puzzle, usually in a program like Chimera or Chimera X. And you can see an example of that over here on the left. That's a structure with the EGF receptor and then its interacting partners there. You then need to bring that structure that you've built into your animation software, which we can see in the top right here. The software that I work with is called Maya, although I am working with Blender now as well. I'm starting to learn that. And what you can see here is this horizontal series of structures over on the left in the animation software here over on the right. But they're below the cell membrane there, which is this coral-like structure. And this umbrella-like structure at the top is above the cell membrane where you can't see it. You then have to rig and animate your structures. And rigging typically involves adding a skeleton of joints and bones to articulate the structure in much the same way as your own joints and bones articulate your body and move you around. You can then apply forces to that rig to animate the molecules and make them move around. And throughout that process, you will perform lots of test animations called play blasts to ensure that the animation is unfolding in the way that you like, that things are interacting properly and that the timing is good. So I'll just show you a little example of a play blast here. And so you can see that's the cell membrane there that you can see above, which is being controlled by that lattice structure there. you've got those two molecules interacting, there's a chain, and then they part. And so you will be doing lots of these throughout the animation process to ensure that things are working the way you want them to. You will then texture and add pretty colours to your molecules to make them look beautiful. You'll add lights to the scene so that you can see everything, and you'll add cameras through which you'll be looking at the action you're trying to capture. So it's a lot like being a film director in a digital format. You will then render the animation, and rendering is a process whereby for each second of animation, you have 25 frames, and rendering is the building of each frame pixel by pixel. In my case, I work with 1920 by 1080 pixels. And so rendering is the process of putting all those pixels together for each frame of the animation. You then have to take all those frames and put them together to make a coherent movie, which is a process called compositing. And I do that in After Effects, but you can use Final Cut Pro or Premiere or anything like that. At this point, you will kind of edit as well to make things flow a bit better if you need to. You can add special effects, you can add labels and captions. I will then add sound effects, and I work with two fantastic sound guys called Adam Hunt and Ryan Granger, and they do amazing work punctuating all the movement that's going on. And finally, we'll add a voiceover narration, and I work with Dr. Claire Fideli, who was a scientist for this, and she's amazing. So let's take a look at some artistic considerations that you need to be aware of when you're making an animation. First and foremost, the story determines the design. What you're trying to say, the story, drives the look and feel of each shot, and not the reverse. So you're not trying to make the shots look beautiful at the expense of the story. You may have heard Pixar say, story is king, and this is what you need to keep in the forefront of your mind. Camera movement and lighting are really important for directing the viewer's attention, and you can use camera movement to take the viewer on a journey from point A to point B. So that really directs where the viewer's looking. Choice of colours. This is quite contentious, a little bit controversial, because most molecules are not naturally coloured. There are many that are. For example, our favourite genetic reporter, green fluorescent protein, indigo, which is blue, ferrocene, which is kind of rust coloured, but many are not. And so there are many purists out there who say that it's not scientifically accurate to have coloured molecules in your animations. There's a woman in Italy called Monica Zoppa who works entirely in grayscale, and you should check out her work. It's pretty amazing. Her work looks really beautiful despite being entirely in shades of grey. But I love colour, and it's a signature of my work, and I like to use it to indicate disease states. For example, if I'm trying to talk about inflammation, I will use warm pinks and reds to indicate that. And for example, if I'm animating cell death, I will use murky greens and yellows. And these ideas, they register subconsciously with the viewer. They just seem to inherently understand that this is what you're trying to say. You can also use colour to define the different functional parts of various molecules. For example, over on the bottom right there, on the left area of that image, there is the structure for BRCA1, which is implicated in causing breast cancer. And I just made the various parts of that structure varying shades of sea green because I thought they were beautiful. So it's a completely personal choice in this situation. You can use glows to highlight molecular and cellular interactions. For example, insulin binding to the insulin receptor. That can really punctuate the movement and direct where the viewer is supposed to be looking. And you can use sound effects as well to great effect. And again, these actually direct the viewer's attention and they promote understanding. And they're not an afterthought. If you've ever looked at a biomedical animation or an animation without sound and then compared it to the same thing with sound, it really brings things to life. So it's a very important consideration and is integral to my productions. So I'll just talk a little bit now about how we're actually using animation at Peter Mac. So firstly, we're creating the usual screen-based animations, which you may be familiar with. And what I'm going to show you now is an edit of our latest animation about chimeric antigen receptor T cells, which is a treatment technology that we have at Peter Mac, and it's just recently come on board, and it's really firing the public's imagination. So this edit explains how these receptors are structured on the surface of killer T cells and how these CARs, as they're known, enable the T cells to seek and destroy cancer cells. So let's take a look at that.
In the laboratory, T cells are instructed to produce special receptors called chimeric antigen receptors, or CARs, on their surface, making them into CAR T cells. The CARs consist of different components that occur naturally in cells, but which are never found together. The CARs sit on the surface of the cell and are engineered to recognise and attach to a patient's cancer cells via the binding domain.
There's a glow, you see there?
We will now move below the cell surface. Here, we are looking inside the CAR T cell at the CAR signalling domains.
More glows.
Once the CAR T cell is bound, CAR signalling domains begin a chain of events which activates its killer function to destroy cancer cells. A CAR is therefore a modular combination of cell recognition components and signalling components all in one. We are now looking at the magnified surface of a CAR T cell in contact with a cancer cell. Once they are in the body, the CARs can directly bind to specific markers on the patient's cancer cells, shown in yellow. The bound CARs then cluster together. We will now move below the cell membrane. Here, we are looking inside the CAR T cell at the CAR signalling domains. The binding interaction has activated the signalling domains, beginning a chain of events which activates the CAR T cell. Once activated, the green CAR T cells release toxic chemicals into the cancer cells, which kills them, indicated in red. CAR T cells are known as serial killers because they can kill, release, and move on to kill other cancer cells in a cycle that can occur multiple times. This makes them very effective in the fight against cancer.
You can also use animations as the centerpieces for interactive tablet-based resources, and these can be used in patient appointments. So this particular resource was designed to help a group of lung cancer patients that have a mutation in a protein KRAS, which controls cell division. And this resource helps the patients better understand their cancer biology and to make informed decisions about their treatment, and maybe even whether to participate in trials that are testing new treatments. So you can see over here on the left, it's an iPad-based resource. There is a keyboard and a stylus there. There's welcome information and an introduction, giving you a background to KRAS. There's a video explaining the biology itself. And then there's a series of still images taken from that animation that the doctor can annotate in real time. So you can draw on the images themselves to indicate this is where the treatment actually fits into that KRAS structure to keep the off switch in place to turn off that out of control cell division. Then there's a series of terms and definitions, and then this whole thing can be packaged up as a PDF and sent to the patient. And so we're actually already conducting a study in the clinic where we're using this technology in patient appointments, and we're then interviewing them after they've seen it, and we're going to statistically analyse the interviews and then determine whether this is a good technology for explaining cancer biology to patients and whether we could apply it to other cancers. We also create still image exhibitions from the animations that can be animated with an app called Artivive on your smartphone. So I'll just show that to you now. Hello, I'm Dr. Maya Divyak and I'm the biomedical animator at Peter Mac. We have just recently launched our latest bioanimation called What Goes Wrong in Cancer. And to go with that launch, we have a series of still images here taken from the animation. And these images are actually interactive. So if you use a specialised app called Artivive and you hold your phone up to the image like this, you can see that it comes to life. So you can find this exhibition on level 7 in the atrium, so do come down. So you can see that animations can actually achieve a life beyond the confines of a computer screen. And that's where it gets really interesting. We've also created holograms from the animations. So this allows the beautiful graphics to reach a wider audience in a purely aesthetic form, and they're not meant to be educational, they're meant to be inspiring and beautiful. So the animations are actually projected from a television screen that sits horizontally above a four-sided transparent perspex pyramid. And the images are actually projected into the pyramid, forming a complete sphere inside the pyramid. And so this gives them an actual presence. So you can walk around the pyramid, you can look into it, you can poke it. And so it becomes a quite personal or communal experience, and it's a physical one as well. And these may inspire more questions than answers. We actually tested these holograms at an event called Testing Grounds, which is a forum for artists to test works in progress, and they were really well received. So we're thinking of what we are in the process of actually constructing these holograms for an exhibition at Peter Mac and to also take on the road to various events and conferences and so on. So let's take a look at that. Hello, I'm Maya. This is an installation that I created with Eric Jong, who is the holographer, and I'm the animator. Hope you like it. So there's the Perspex pyramid with the TV above it. You can see the sphere inside the pyramid. That's Eric Jong. He's the artist that developed this technology. I'm collaborating with him. We are testing ground. So let's talk a bit about me. How did I become a biomedical animator? I went right through the academic system to postdoctoral level. So I did an undergraduate degree in biomedical science. I then went on to do honours. I worked as a research assistant for about 3 years. And then I came to Melbourne to do a PhD. Then I worked as a postdoc for about a year after that, before I kind of, came to the realisation that maybe a career in bench-based research was not for me. And I then went into scientific sales for about 10 years. And about the 8-year mark, I started to get really aware that my artistic side needed a creative outlet. I had always been quite artistic at school. I had an even split of art and science subjects. I studied fine art and French in addition to chemistry and biology. And that whole artistic side took a complete backseat when I was going through university and climbing the academic ladder. And it was just yearning to come back out again. But I was really struggling to think of a way of bringing art and science together, my two favourite things. So I was asking, where's the art? And as luck would have it, I met a wonderful man called Dr. Drew Berry, who's probably considered the world's leading biomedical animator. And he works at the Walter and Eliza Hall Institute, and I was there for my sales work, and it was just one of these chance meetings. And he showed me his work, and to me, it just seemed the perfect combination of science and visual representation. And that was the aha moment for me. And I said, yes, this is what I want to do. So I set about retraining to become an animator. I went to Swinburne and studied multimedia and design. And that allowed me to create enough material to put together into a portfolio to apply to the Australian Film, Television and Radio School, where I did an intensive 3D animation course. I then set about doing a lot of work on my own, trying to get my skill base up, doing a lot of freelance projects, animating my PhD, which was a really great piece of advice from Drew, because you know it so well and so intimately. Just really trying to get my skill base up and getting a really good portfolio under my belt. I was then really lucky to be offered an 18-month fellowship with Drew at WeHi, and that's where I found the art and where I learnt most of what I now know. I got my first job as science animator at the Gene Technology Access Centre, which is on the grounds of the University High, which is right next door to WeHi. And I'm currently biomedical animator at Peter MacCallum Cancer Centre, as you well know. And I actually went to the executive director at Peter Mac and I said to him, this is who I am, this is what I do, and this is why you need me. There was no job. I had to actually collaborate with them to make a job in this field. So you might ask, well, how can I become a biomedical animator? First and foremost, you have to be really interested in science. And in all reality, you'd probably have to study to at least master's level in a biology-related field to be able to have the skills to interpret the data and to be able to represent it visually. That is, of course, unless you work in a studio overseas where the information is provided for you and you are told to animate something. But of course, if you have science and you understand it, then you will make a much better science animator. Then, of course, you will need to study 3D animation. And unfortunately, there are no specific biomedical animation courses or even a well-defined career path in Australia for the entertainment industry and visual effects. In particular, there are amazing courses at RMIT. There is plenty of online material for biomedical animation that is specific to biomedical animation. So it just depends on how you like to learn, whether you like more formal structured courses or whether you're more self-directed. And if anyone wants any information about any of these offerings, any links, so on, I'm really happy to share them with you. There are actually fantastic courses in Canada and the US that are specific to biomedical animation, but they're very competitive and you will need a pretty strong portfolio to apply. But of course, someone has to get in, so why not you? As mentioned, the career path in Australia is a bit sketchy. There are a few job opportunities, but there are many internationally. If you want to stay in Australia, you'll probably have to forge your own career path in the way, sort of the way that I have done. And that will probably involve freelancing to begin with, which, as you know, means there could be periods where you have so much work you don't know what to do with it, and periods where you don't have anything for months on end. So it can be a difficult existence, but it will be great for getting your skills up. You could also study or work overseas, as mentioned, to gain valuable experience. This is also really good for personal development. And something that I'm seeing more and more now is that a lot of international scientific animation studios are offering remote work to international employees. So this is actually opening up the market a lot. So although there are a few opportunities in Australia, this actually gives you the chance to be part of a developing movement and to shape the way that you really want to work. So you'll be at the forefront of a revolution in scientific realisation if you choose to study and stay here. I can see a situation in the future where scientists applying for grant funding will have to have a visual component explaining what they've discovered at the end of their research process. So I can see visuals becoming more common in the science world in Australia. So I think if you were to get into this field now, you would be well placed. So back to those perceptions of science. We started out with science is boring. And then we went on to science is complicated. And finally, scientists are nerds. With biomedical animation, I really hope that I've convinced you that it's a wonderful tool for making science interesting, for enabling understanding and therefore making science accessible, and that maybe being a science nerd is not such a bad thing. I love what I do. And if you're interested in science and representing it visually, you will love it too. Above all, I really hope that I've made you aware of the inner workings of your cells and that what happens there is extraordinary. Thanks so much for your time. These are my acknowledgements. I won't go through them now, but I would really like to thank the Peter Mac Cancer Foundation for support of what I do. I am paid entirely through philanthropic funding. You can reach me on my Twitter handle or my email address. As I mentioned, please contact me if you'd like further information. And yes, the intersection of art and science is indeed a place of wonder, and I'm so happy that I inhabit that nexus on a daily basis. Thanks so much.